Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class we discussed about cytosine methylation and different methods that could detect the methylation on cytosine. We also talked about different methods that could distinguish between two different types of modification on cytosine, for example, the methylation, right, and the hydroxymethylation. So, we talked about different methods that could do that. So, one of the questions that still remain, how do we actually identify the methylation pattern on adenine, right. So, on A base. Okay. So, this is something that we have not talked about so far, and it turns out this was not so focused on because these were actually not very common in the human genome. Okay. So, in this class we will talk about methods that could allow us to identify these 6 MA modifications or 6 MA bases, and then we will talk about different methods that could actually help us identify the transcription factor binding sites across the genome. So, this is something that is very important because all these methylations actually impact the binding of transcription factors, as we will see in this presentation. Okay, so, these are the two topics that we will be discussing in this class. So, direct detection of DNA methylation patterns that would actually allow us to identify the 6 MA bases, and then we will talk about transcription factor binding site identification using a method called chromatin immunoprecipitation or CHIP. These are the keywords for this class: interpulse duration, pulse width and nanopore, and then we will also see immunoprecipitation. So, just to summarize what we know about DNA methylation so far, right. So, these are methylation on A or C base mostly. Of course, we have talked about some other modifications as well, and we have talked about detection of 5 MC and 5 HMC bases. These are modifications on cytosine, specifically talked about bisulfite sequencing that could actually easily allow us to identify these modifications at the genome level, so across the whole genome. And we also talked about TAP-seq and OXPS-seq. These actually allow us to distinguish between 5 MC and 5 HMC modification, because these are important for certain applications. So, the question now is, how do you detect methylation on A bases? Okay, this is something that we want to identify, and just to remind you, this is the modification that we see on A is called 6 MA. So, the modification on A is very common in prokaryotes, and it's thought to be uncommon in eukaryotes, especially in mammalian cells. However, recently papers have shown that these 6 MA methylations are also present in human genome, and they actually mark actively transcribing regions, and these modifications levels of these modifications change in human diseases. So, here is the reference. If you are interested, you can go and read further. So, how do we detect these 6 MA bases in the genome? So, it turns out we have to resort to something called direct detection of DNA methylation, and there are two sequencing technologies that actually allow us to detect this DNA methylation directly. Okay. So, remember, right, in bisulfite sequencing we need to do a treatment with sodium bisulfite, and this is a quite harsh treatment for DNA. You can have also other sorts of damage to the DNA and other bases. So, that if we can avoid that, right, and if we can go about detecting these methylation patterns just by sequencing, that would be really ideal. Okay. And it turns out two methods actually offer that opportunity. The first one is single molecule real time or SMRT sequencing, and the second is nanopore sequencing. So, we will discuss little bit about these methods and how they allow us to detect this DNA methylation directly from the sequencing data. Okay. And these two methods, what is common between them is that they are single molecule sequencing, right. So, they are sequencing single molecule, and we also get the real time data, okay, unlike the earlier methods. Okay. So, let us now see how we can apply these methods and how these have been applied to actually identify DNA methylation in practice. Okay. So, first we will talk about SMRT sequencing and how we can apply this method to detect DNA methylation. So, if you remember SMRT sequencing, the sequencing happens at the bottom of the zero mode waveguides or ZMWs, right. So, you have the DNA polymerase attached to the bottom of those ZMWs and the DNA molecule comes. Okay. So, the polymerase is there and the DNA molecule comes and the sequencing of the complementary strand, the synthesis of the complementary strand happens and the nucleotides come and bind, okay, and these nucleotides are attached with fluorophores. Okay. And this means when the nucleotides come and attach to the DNA, they are held in place by the polymerase and this is where the signal detection happens. Okay. Now, since this is a single molecule real time sequencing, the time of addition of this individual base is recorded in the raw data. Okay. So, you can find out this data, right, at which time points these additions are happening. Okay. And then if you know the time, you can look at the interval between two base addition events. Okay. And you can also look at another parameter which is the time of or the duration of signal, right. So, for which the time for which you can actually detect the signal. Okay. So, these are the two parameters, the technical terms for those. The first one is pulse width. So, pulse is addition of base, that is the event, right, and the width of that pulse or the time interval for which we can detect the fluorescent signal, right. So, let us say you are adding an A base and you can say the detector can detect this A base fluorescence for let us say a few milliseconds. So, that would be the pulse width and the second is interpulse duration or IPD. Okay. So, this is the time interval between two consecutive pulses. Okay. So, you have let us say addition of base A and then addition of T. Okay. So, the time interval between these two base addition events is called the interpulse duration. Okay. So, you can then observe this interpulse duration for the full process, okay, and this data is stored in the raw data. So, using these two data sets, now we can actually see, right, if you have methylation, there is increase in interpulse duration. So, here is the paper that actually looking at direct detection of DNA methylation using this SMRT sequencing, and what they observed is that if you have methylation on A, for example, you can see this difference in interpulse duration, there would be an increase in interpulse duration. So, if you have a base and there is methylation, the time taken would be more for the next base to come. So, we of course, do not understand why that is the case, perhaps this is something to do with steric hindrance, the structure of the whole thing, etc. So, this turns out that this method can detect 6 MA as well as 5 MC and 5 HMC, right. So, you can imagine, right, these chemical structures are different. So, they affect the signal in different ways, right, or these parameters in different ways, which we can then identify for identifying these methylation patterns. Okay. And what we look at is the IPD ratio between sample with methylation and the control. This is what this paper actually did, and they actually looked at this modification, that certain specific genomic context or specific sequence context, and then looked at these IPD ratios between sample with methylation and the control without methylation. And what they observed that for 6 MA there is an increase in this IPD ratio, and this increases at the position of the methylation. Okay. So, where this methylation is present, there is an increase in the IPD ratio in the sample, right, compared to the control. Now, for 5 MC this IPD ratio increases at second, third and sixth position after the methylated bases. Again, we still do not understand, right, why that is the case, how they influence this like IPD values at these positions after the methylation, but this is what they observed. Again, this is with respect to a specific sequence context. So, it might be the case that if you change the sequence context, these values might change. So, again, before you actually apply these methods, you probably have to optimize and look at these controls. For 5 HMC, there was a small difference. So, the IPD ratio actually increased at second and sixth position, but not at the third position after the 5 HMC base. Okay. So, what they could do is actually also differentiate between unmethylated C, 5 MC and 5 HMC bases based on these two parameters that we just described. So, this IPD and pulse width parameters, if they used they could actually cluster out these unmethylated cytosines, 5-MC and 5-HMC bases, okay, and they could see these different clusters based on which they could identify the modification. But what I have just mentioned, this is dependent on the sequence context, right. So, if you change the sequence context, it is likely that the signals, these IP ratios, etc., these will change. Now, let us move on to the nanopore sequencing, how you can apply nanopore sequencing for detecting DNA methylation. So, okay, so if you remember, in nanopore you have these channels, right, through which the DNA molecule passes and it actually changes the current, right. So, there is a current at the picoampere level going through the nanopore, and the moment DNA enters, right, it changes the resistance, and that actually alters the current, and this change in current signal is detected by the detector, and then the base call happens. Okay, we talked about this in detail in the beginning. Okay, so what happens is that if you have DNA modifications, right, these are actually unique signals, chemical signals, right. So, they actually induce distinctive changes in the current, right. So, the moment you change the chemical structure, the resistance will be different, that will induce distinctive changes in the current, and this actually has been used for distinguishing A and 6-MA as well as distinguishing C, 5-MC and 5-HMC bases. Okay, so, here are two papers which are looking at this detection and mapping with nanopore for these different types of modifications. Okay, so, you can go and look into their data, right, and how these current signals are different from each other. Now, what we have learned about DNA methylation detection is that traditional methods for detecting 5-MC, 5-HMC require chemical treatment of DNA, and that might be harsh in some cases. It also increases the experimental complexity as well as the data analysis complexity, right. So, if you have to do this 3-way comparison between samples, right, that actually increases the data analysis complexity. What we have seen is that SMRT and nanopore sequencing, they actually enable us to directly detect these DNA methylation patterns, and they can differentiate between different types of methylation patterns. But of course, the accuracy is not actually like perfect, right. So, there is a lot of scope for improvement in these direct detection methods, but nevertheless they provide direct identification methods. Now, why are we interested in detecting these DNA methylation patterns, right? So, one thing we have learned is that these methylations are important for influencing gene expression. For example, we have seen that methylation of C residues in CPG islands in promoters of genes can silence transcription. Okay, this has been observed for a long time, and what we know is that this methylation actually inhibits binding of activators that will induce gene expression, or it attracts repressors that actually blocks this expression. Okay. So, this means this DNA methylation actually is intricately linked with gene expression. Okay. So, they are controlling gene expression. So, can we identify these sites of transcription factor binding in the genome directly, right? So, instead of relying on this methylation information, right, can we directly identify the binding sites of transcription factors in the genome? Okay. So, ultimate goal is to understand how epigenome regulates gene expression, right. So, if we can identify these transcription factor binding sites and see whether transcription factors are bound to specific sites, and then we can correlate that with the gene expression levels. So, as I said, this actually will influence gene expression, and then of course, we can also connect this information with the DNA methylation studies, right. So, we can then say, okay, this kind of methylation pattern blocks these transcription factors, or this kind of methylation pattern attracts this kind of transcription factors, right. So, this is something that will be more informative in association with the DNA modifications. Okay, so, we will talk about this method. It is called chromatin immunoprecipitation, that actually allows us to do so, and it can be combined with next generation sequencing, and this method is called chip-seq, and earlier it used to be applied with microarray technologies, and it is called chip on chip. Okay. So, these are the full forms, right. Chromatin immunoprecipitation chip with sequencing, and chip on chip would be chromatin immunoprecipitation with DNA microarray. Okay, so these methods actually allow us to detect binding sites of transcription factors in the genome, and it can also detect binding of other DNA binding proteins such as chromatin remodelers. So, any protein that can bind on the DNA, we can detect their binding sites, their locations, etc. And we can also detect the sites of histone modifications. Okay, so we have not talked about this at all, and ChIP-seq offers us a way to actually identify these histone modifications. Okay, so what are the steps? So the first step is actually cross-linking of DNA and protein using formaldehyde. Okay, so this actually preserves this connection between DNA and protein, right. So at certain sites we have protein bound to the DNA, and we want to preserve those connections, right, before we actually proceed for DNA fragmentation, etc., because otherwise these connections might get lost when we are preparing the sample. Okay, so the first step is cross-linking, where we preserve the connections that we want to study. The second step is DNA fragmentation, right. So we create smaller fragments of DNA. The third step is immunoprecipitation using antibody, right. So this antibody will be targeted against the protein that we are interested in, or against histone modification that we want to identify. And then the final two steps, these are very familiar to us. The first is library preparation, and the second is sequencing. Okay, so let us move on to this animation, so you will understand this process better. So what you have is this DNA, right. So you can see these histones and their modifications on top, right. So for some histones you have methylation, some acetylation, just for illustration purpose. And then you also see certain proteins that are also bound to DNA. These could be transcription factors or other proteins, right. The process remains the same. Okay, so what we do is we do the cross linking, right, and then we have the fragmentation. Okay. So once we have this fragmentation, okay, we get the fragments and we do the immunoprecipitation using antibody. Okay. Now we can design antibody that will detect these specific methylation patterns on histones, or we can design antibodies against these proteins, transcription factors or chromatin remodelers, or any other protein that binds DNA. Okay, so once we have this immunoprecipitation, so what happens is that we are pulling down these regions of genome, and the rest of them are washed away, right. So these regions are washed away in this process. So once we have pulled these regions down, we then purify the DNA fragments and then subject them to next generation sequencing, so that we identify these specific regions where we have this DNA binding of event, right, whether it is histone modification or transcription factor binding. Okay, so the next step is we want to identify the binding site from read data. Okay, so here is the challenge. Okay, when you have isolated this DNA fragment, right, and sequence them, the reads will appear from the end. Okay, so we sequence from either end, right. We can use pair end sequencing and sequence from both ends. Okay. So the reads that I am showing in blue color here, right, they will mostly appear at the end. So these regions we cannot control the size of these regions, right. So they could be quite big, could be 1 kb, could be even longer, right. So we cannot control the size, but then remember the sequencing method is if it is Illumina, it is short read sequencing traditionally, right. So the reads will be concentrated at the end of this DNA fragment. Okay. So similarly in this case, if you are interested in determining the binding site of this protein, right, will be left with these reads that are coming from the end of this DNA fragment. So the problem is we have to now determine the actual binding site from the distribution of reads here. Okay. So the actual binding site is somewhere in between these paired end data, right. So between these two sets of reads, somewhere in between, but from the distribution of reads we have to infer the binding site. Okay. So this is an inference problem, we cannot directly determine the binding site. Now the question is, what is the exact location of the tape binding site or the histone modification, okay, that we are interested in. So there is a computational analysis, and this is called peak calling. I will not go into the details. So the idea is looking at the read distribution pattern, we have to infer the actual binding site. Okay, but then again this is an inference or prediction based on the distribution and it is not actual observation. Okay, so this is what this method will do. It will determine the binding site or site of modification, and the resolution depends on the fragmentation sites. So again, these fragmentation sites are not controlled in most cases, right. So they happen randomly, and this resolution is dependent on the fragmentation sites. Why? Because if these fragments are small, right, then the reads will come close to the actual binding site, and in that case inference would be better. But if the fragments are large, the reads will be concentrated towards the end of the fragment, and then inference will be much more difficult. Okay, so this resolution is usually in 50 to 100 base pairs, but the actual binding sites of transcription factors, they are even less than 10 base pair to up to 12, 15 base pairs. Okay, so you can see that there is a huge scope for improvement using this ChIP-seq method. Now, we also do something called differential binding analysis. So, this is comparative analysis between two or more samples, and this is focused on changes in binding sites or sites of modification. The process remains the same, is that we are just doing this for two different samples and then we are comparing, right. So, this could be very useful if you want to compare these transcription factor binding sites between two different conditions or between diseased and healthy samples, and very similar to what we talked about in case of transcriptome analysis, right. So, we talked about differential gene expression analysis, and similarly we can do differential binding analysis for transcription factors, and then of course, we can connect this to the transcriptome data, right. So, whether these differences in binding of transcription factors can influence the gene expression pattern. Now, one of the questions that we have is that can we determine the peaks with higher resolution. So, as we have seen, ChIP-seq actually does not give us the desired resolution. So, can we determine the peaks with better resolution? Okay. And then we can then identify or determine the actual binding sites, not just infer or predict, but we can actually see the actual binding sites, and we can do something called motif analysis for binding sites. So, what are these motif analysis? So, this is motif is a specific order of bases recognized by a transcription factor. So, for example, a transcription factor recognizes these bases or this sequence A, G, T, A, G, A, T, and each transcription factor has a signature sequence or maybe sometimes more the signature sequences that it can bind to, right, because of the chemical interactions, etc. And if you know this motif sequence, you can then search across the genome for the binding sites or putative binding sites of that transcription factor, right. So, this is actually very useful if we can determine the actual binding sites. Okay, and this method was proposed to do that. It is called ChIP-exo. So, this is chromatin immunoprecipitation with exonuclease treatment. Okay, so, the steps are mostly similar. So, you can see first three steps are exactly identical as before. So, we have this cross linking, then you have fragmentation, then you have immunoprecipitation using antibody, but there is an extra step here which is the exonuclease treatment of the purified DNA. Okay, so, or the purified fragments that we get. Okay, and then we have library preparation and sequencing. Okay, so, what happens is that after immunoprecipitation, once you take out these fragments or pull down these fragments, then we subject them to exonuclease treatment. So, these are the exonuclease enzymes animated, and what they will do, they will chew away these bases from both ends. Okay, so, they will chew away the 5' ends, okay, and what will happen is that you will get much smaller DNA fragment. Of course, they cannot chew the sites that are bound by these proteins, right. So, what will happen is that they will stop once they encounter these proteins. Okay, so, you are left with these very small fragments, and now if you sequence from both ends, you will get reads mostly focused on the binding sites. Okay, so, you do not have to infer these peaks anymore, right. You can simply directly identify the peaks from this data, and this is a much direct and simpler peak calling method. Okay, so, from ChIP-exo we get high resolution peaks and sometimes even at single base resolution. So, this is great. We can identify the actual binding sites. We can then do the motif analysis for transcription factors. As I mentioned, we can identify the specific set of bases that transcription factors identify, and then we can also look for putative binding sites of those transcription factors across the genome. Now, there are several factors that actually affect quality of ChIP-seq and ChIP-exo experiment. As you can see, these are quite complex experiments, right? You need an antibody, then you have pull down, then you have, if you are going for ChIP-exo, you need to do the exonuclease treatment. So, this is quite complex, and we have to be aware of the factors that can affect the quality of the data. So, the first thing is the antibody quality and specificity. As I mentioned, this is very critical for the process, and also the sample quality. So, the antibody quality is very important. It has to be very specific for the protein that we are interested in. If it also binds to other non-specific targets, those will also appear in the results. Okay. So, this is something that we need to take care of, and we do so by having control experiments. Okay. So, these are experiments that we also do along with the original ChIP-exo or the actual ChIP-exo or ChIP-seq experiments, okay, just to ensure that the results that we get are robust and accurate. So, there are three types of controls that we need. The first is input DNA control. This is prior to immunosuppression. So, remember what we will get after ChIP-seq or ChIP-exo. We will get reads mostly focused or mostly from specific regions of the genome, right. So, if you are looking at, let us say, transcription factor binding sites, we will get reads mostly around that region. Okay. So, there should be an enrichment of reads around that region, and that has to be compared with the input DNA control, right. So, because this enrichment could also happen in random, right, because as you can see, the coverage could fluctuate across the genome. So, this fluctuation we also need to consider when we are analyzing or looking for enrichment in case of ChIP-seq or ChIP-exo, right. So, that is why we also need to have input DNA control. This is prior to immunoprecipitation. Okay. So, whatever we have prepared before we add the antibody, we also take those fragments and sequence. Okay. So, it is before immunoprecipitation and after immunoprecipitation, we see the enrichment, and that gives us the transcription factor binding sites. In addition to control for other non-specific bindings, right. So, we also do ChIP experiments from mock immunoprecipitation. So, this is DNA from mock immunoprecipitation without any antibody. Okay. This is again looking for biases in the sample preparation protocol. And the third one is DNA from non-specific immunoprecipitation. So, this is non-specific antibody that should not actually lead to enrichment of any kind in those specific regions, right. So, we also include that ideally, because this will tell us, okay, how much of the enrichment that we see is occurring by chance. Okay. So, these three type of controls would be ideal for any ChIP-seq or ChIP-exo experiments. Here are the references for this class, and to summarize, we have talked about direct detection of DNA methylation patterns and using two different next generation sequencing method, the single molecule real time sequencing or SMRT sequencing and nanopore sequencing. And we have seen that they apply two different principles to actually identify these methylation patterns. The SMRT utilizes two parameters, which is interpulse duration and also the pulse width. So, based on the distribution of values for these two different parameters, the methods could allow to distinguish between these methylated bases. And nanopore sequencing, it looks at the current signals, and apparently each molecule or each modification has a distinctive influence on the current signal, which could then be utilized to identify the modification. We have talked about chromatin immunoprecipitation. This can identify binding sites for specific transcription factors or specific histone modifications. So, if you are working with antibody against transcription factors, we will be looking at binding sites for specific transcription factors, but we can also design antibodies against specific histone modifications, and then we also know the histone modification sites in the genome. We can apply ChIP-exo, ChIP-seq and ChIP-chip, right. So, we have talked about this ChIP-exo and ChIP-seq, and if we talk about resolution, ChIP-exo has higher resolution than ChIP-seq and ChIP-seq has higher resolution than ChIP-chip. This is something that is seen in the data across different samples. And one of the major challenge to ChIP experiment is obtaining good quality specific antibody. And as I said, right, you need to have antibody that is specific against the target. If you have non-specific interactions, that can bias your results. So, this is a challenge for these experiments. So, we will talk about other different types of methods that can actually allow us to look at these different binding sites or look at the nucleosome occupied sites in the next class. Thank you very much.